So, virtual attendees, hope you all are doing well and staying safe. I am Anoop Kepal and I am the Chief Operating Officer at IZON Science US Limited. I am excited to share some of the recent product developments at IZON Science. These developments are focused on the EV community's long cherished need for standardizable and reproducible methods. Most of you would have seen this figure and it aptly summarizes the excitement surrounding potential of extracellular vesicles as well as the hidden complexity of these bioentities. Now, tremendous interest does lie towards development of EV-based biomarkers for diagnostics and prognostic applications. Several studies have shown the utility of EV-based biomarkers, especially microRNAs, for monitoring disease progression and response to therapy. Broadly, EVs are membrane-bound vesicles that are very heterogeneous in size, number, membrane composition, and cargo. Due to this, it's a challenge to isolate, purify, and further characterize these different types of EVs to eventually evaluate the impact of biophysical properties on functional outcomes. Moving on, let's have a quick look at what it means in terms of EV cargo complexity, especially RNA content and different xRNA classes. This figure reflects upon different cellular processes that result in release of microRNA from various cell types into the bloodstream. Apoptotic cells, which is apoptosis, is programmed cell death that results in release of RNA transcripts and short DNA fragments along with cellular remnants and apoptotic bodies. In contrast, necrosis is more of a random process and results in release of long DNA stretches. In both these cases, the RNA transcripts bind to specific RNA proteins, for example, agoproteins. EVs, on the other hand, are more of active cellular secretions. These circulating vesicles are thought to contain cell-specific microRNAs that are involved in cell-to-cell communications. In summary, the two major current challenges in the EV field are: one, lack of standardization when it comes to methods of EV isolation and characterization in different biofluids, and second, how to resolve EV specific signals from background noise. That is, how do you separate vesicular particles from non-vesicular particles like lipoproteins and RNPs, that is ribonucleoproteins. Now a related question is, how do you balance the requirement of high purity vesicles and still get enough yield for different downstream assays? These are important considerations for EV-based biomarker discovery projects, and once a robust biomarker is found, how easily the same method could be scaled up for evaluating larger cohorts of samples, generally required for bigger clinical studies. At IZON, we have developed methods to simplify the overall EVV workflow from isolation to characterization and further downstream analysis. Using size exclusion chromatography based QEV columns, one could easily purify different biofluids like plasma, cell culture, urine, and obtain highly purified EVs. The resulting EVs are intact and can be further used for characterization. So one could actually do size, concentration, and surface charge analysis. These intact EVs could be further used for undertaking functional activity assays for therapeutic evaluations. As required, the EVs could further be concentrated and lysed for content evaluation, including RNA analysis, proteomics, and lipidomics. On this slide, I have summarized some of the typical methods for EV isolation. Ultracentrifugation is one of the common methods and has been used for a long time, but has limited utility when it comes to processing large number of clinical samples. It is time consuming and labor intensive. It requires use of high centrifugal forces that could compromise structural integrity and biological activity of exosomes. Precipitation kits, on the other hand, lack specificity and result in contaminated EV preps. Size exclusion chromatography columns, or SEC, is based on size separation and is independent of binding affinity. This method is overall gentle, repeatable, and fast. I have shown I have shown few publications here. There are several independent publications which have compared different EV isolation methods and have pointed out that SEC purified EVs are biologically active and show higher functionality in comparison to other methods. SEC purified EVs are structurally intact and devoid of non-vesicular contamination typically observed with precipitation kits. To go over the SEC process very briefly, the column matrix or gel has pores of known sizes, and in our case it could be either a gel with 35 nanometer pore size or a gel with 70 nanometer pore size. Once the sample is overlaid onto the column, larger particles they pass through the pore faster and elute earlier, whereas the smaller particles they tend to take more of a tortuous path and elute off in the later fractions. In the figure which is shown here, the blue line is indicative of smaller proteins and non-vesicular components, whereas the red line is actually for EVs, which are eluted off here. And before that, the volume is actually the void volume. A typical sample run takes about 15 minutes, and the resulting EV preps are devoid of contaminating background proteins. At present, QEV columns are offered in 5 different column sizes and 2 different gel types. QEV singles, as the name suggests, are designed for single use and have 100 microliter sample load volume capacity. QEV originals have half ml sample load capacity, and QEV2, QEV10 and QEV100 have 2, 10 and 100 ml sample input volume requirements. In terms of gel types, 70 nanometer gel type separates particles in 70 nanometer to 1000 nanometer range and results in high purity of EV preps, whereas 35 nanometer gel type separates particles between 35 nanometer to 350 nanometer. 35 nanometer gel predominantly separates particle below 110 nanometer and shows higher overlap with smaller lipoproteins, especially those that are in 35-40 nanometer size range. Overall, it has higher recovery for smaller EVs. All these columns are manufactured according to ISO 13485 requirements. SEC-based columns are rapidly becoming the new gold standard method of choice for isolating EVs from clinical samples. In a survey that was done in 2015 and repeated in 2019, SEC was reported as the method of choice and that its use saw the biggest increase among the respondents. Noticeably, ultracentrifugation saw a slight decrease. Our own data on the right hand side shows that the EV researchers have well accepted the QEV columns, and the column use has grown 3 to 4 folds over the last few years. A major switch happened when AFC was launched in early 2019. AFC, or Automated Fraction Collector, was developed to streamline the QEV operation, keeping in mind the typical requirements for clinical studies. Except QEV100, AFC can be used with all other QEV types. It has an inbuilt mass sensor that can precisely weigh down to 30 microliter EV volume. QEV in combination of AFC provides a convenient, reproducible, and standardizable method for EV purification from complex biofluids. All QEV columns are RFID tagged, and the users are able to retrieve information about the column type as well as keep track of how many times the column was used in case it was reused. As illustrated in the map, owing to the ease of use and availability of different range of columns that suit the needs of EV researchers, we have rapidly seen acceptance of QEV AFC methodology globally. EV researchers worldwide are quickly moving away from suboptimal and time-consuming methods of EV isolation to much simpler and time-efficient QEV AFC method for undertaking large-scale clinical studies. In a nutshell, one could easily process and isolate EVs from any biological fluid of choice and go on to characterize size, concentration, and surface charge using IZON's tunable resistive pulse sensing technology. Now post column, some of the downstream application assays require further concentration of purified EVs, and that typically requires use of spin filters. Spin filters, as you would all know, could result in unintended EV losses as well as increased variability in the final concentrated output. Based on customer requirements, we at IZON have further expanded the QEV AFC workflow to offer a streamlined and optimized methodology for EV concentration and subsequent EV RNA analysis. I will talk about these kits in the next few slides. These new kits, including QEV concentration kit RCT01, was developed in partnership with Northern Biotech. This kit is optimized to concentrate EV volume between 600 microliter to 400 ml for further RNA extraction. It will allow users to process up to 50 samples and will be available at a price of $750 USD per kit. A different concentration kit, RCT02, has been developed in partnership with Series Nano. This kit is developed to concentrate EVs for Western blots and proteomics analysis. The kit has been optimized to concentrate EV volume ranging from 150 microliters to 200 ml, and the kit is expected to be released sometime next month and will be available in 2 ml and 10 ml slurry format. Another product that we are excited to launch in partnership with Norgen is the QEV RNA extraction kit RXT01. This QEV RNA extraction kit is optimized to process up to 50 QEV purified samples and is available at a price of $394 USD per kit. The kit has a requirement of 600 microliter EV volume input, and for QEV single columns, users could simply use the kit without having to concentrate the resulting EV volume. For processing EV volume between 600 microliters to 4 ml, it's required to first use the QEV concentration kit RCT01 and then concentrate the volume down to 600 microliters. These kits are simple to use and do not require any special instrumentation, precipitation reagents, phenol, chloroform extraction, or protease treatment. The kit is perfect solution for further downstream analysis like qPCR, microRNA analysis, and RNA sequencing. Just to give you flavor of what is in our developmental product pipeline, our team is working on developing a 1 liter input capacity column. This column would allow users to process anywhere between 510 liters of TFF concentrated cell culture media. This column, these are customized for groups looking at therapeutic evaluation of EVs. We are further developing a dual mode sec column to separate out EVs and small lipoproteins in tandem. Here, following a size separation step, a cationic exchange step will be used to capture positively charged LDL and VLDL particles. Similar for the groups working at the interface of EVs and viruses that tend to be of similar size, we are developing a dual mode separation based on size and charge. Additionally, use of virus and EV specific affinity baits are being tested following the QEV step. Thank you for your time and attention. Please do not hesitate to reach out with any questions or comments. My email is anoop.pal@izon.com. Thank you.